Okay, so welcome everybody. It's great to see so many of you here. I'm Alex Pickering. I'm a senior scientific programmer in the Department of Biomedical Informatics, as well as the lead developer on Selenics here at HMS. And I'm going to be hosting the workshop for today. And I just want to start off by um, thanking and acknowledging everybody else that's a part of the Selenics team. I'm just one cog in a much larger wheel. Um, in terms of the structure of the workshop for today, we're going to start by making sure you can all log in and access the workshop data set. After that, I'm going to give sort of a very high level overview of how I think about the main benefits of single cell RNA-seq. Um, and I think that will give you a nice structure for thinking about how Selenics analyzes your data. And then we're going to jump into the hands-on portion of the workshop, where I'll also fill in some of that structure so that you can get a bit of intuition for each of the steps of a single cell analysis. So let's start by making sure you can all log in. So if you could all please, preferably in Chrome, go to selenics.hms.harvard.edu. And please go ahead and try to sign in using your Harvard key. And if you're having any difficulty signing in with your Harvard key, some people it might not let you in based on the role associated with your account. Then please go ahead and post it in the chat. And Genoa will provide you with some temporary credentials that you can use for the workshop today. And once you have managed to sign in, please go ahead and type "I'm in the Zoom chat," and this just lets me know that we're not leaving anyone behind. Um, and if you're either signing in with those temporary credentials or you previously had a chance to log into the platform and add the workshop data set, it sort of was outlined in an email that Genoa sent out, then you're good to go for now. Just hold tight for a minute or so. But if you didn't get a chance to add the workshop data set, please go ahead and do so now by making these 4 clicks down here. So you're going to click, after you sign in, you're going to click Create New Project, then select from Dataset Repository, and then you're going to click the Explore button once in the Dataset Repository. And then finally, when you're back on the main page, in the top right, you're going to click Go to Data Processing. So I'll give you guys maybe just a minute or so in case you haven't had a chance to do that yet. Um, and again, if you're having any trouble logging in, please post it down in the Zoom chat. Great, and I see I finally found the chat. I was looking for it, but I see a lot of you are posting that you're in, so that's great to see. Okay, great. So while you guys are, in case anybody's still working on that, please continue. But while you're doing that, I'm just going to give my very brief high-level overview of how I think about the primary uses for single-cell RNA-seq. So I like to frame this in terms of 2 categories of analysis. So the first is non-comparison based. So these are things that you could still do, even if you have just maybe a single sample, or maybe you do have multiple samples, but they're all from the same group or population. So in that case, you could still run, for example, a trajectory analysis, which would be appropriate if you have a developmental or differentiation data set. You could also look for rare cell types in your sample, or more generally characterize the cell type composition of your sample. So this might be useful, for example, if you're trying to validate an organoid protocol or generate a cell atlas. If you do have multiple samples from different groups or populations, then this opens up comparison-based analyses. So this is your classic disease versus healthy or treated versus untreated type of comparison. And this is really the most common type of analysis that gets performed. So that's going to be our focus for today. And almost always, one of the end results of this type of analysis is what we call a differential expression analysis. And what this does is it looks at each gene individually, and it asks whether it differs in expression between your comparison groups. So, for example, is interferon gamma differentially expressed between our treatment and our control samples? And you repeat this question for each gene in your data set. And this question is really the same whether we're looking at a single cell RNA-seq data set or a bulk RNA-seq data set. So why don't we just use bulk RNA-seq? It's certainly much cheaper. And one of the obvious reasons is that we're interested in knowing what genes are differentially expressed in specific cell types. So for example, maybe interferon gamma is upregulated in our patients' CD8 T cells, but not their monocytes. And we're interested in that context. Another less obvious benefit is that single-cell RNA-seq lets us distinguish between differential expression and differential abundance, which is just a difference in the proportions of your different cell types. So let me zoom in on this second one with an example, just to make sure you understand. So this right here is a single cell PBMC data set that I worked on, where I'm showing you all the cells from two of the healthy samples, as well as two of the patient samples. And one of the things that came out of this analysis is that there was a large difference in the proportion of CD4 naive T cells, so much fewer of them in the patient samples. And if this analysis had instead been run as a bulk RNA-seq experiment, so basically taking all these different cell types for a given sample and blending them together in order to get a single expression profile per sample, then I would have expected this observation up here to show up as differential expression. For example, through a downregulation of genes related to CD4 naive T cells. But I doubt that I would have been able to actually come to the correct conclusion about the reason for that downregulation. So that's a benefit that we get with single cell RNA-seq. Let's come back to this first point and look at exactly how Solenix does this cell type specific differential expression, because as I mentioned, this is really one of the main outcomes of our analysis. So the way that Solenix does this is it picks a cluster For example, these CD14 monocytes, and it separates them by sample. So our two healthy samples in this data set and our two patient samples. And then for each one of our samples, it takes those monocytes and it applies our digital blender to them, which is just the sum function in this case, in order to generate what we call a pseudobulk expression profile for each of our samples. So for example, we're taking the 212 monocyte expression profiles from this healthy sample. We're summing across genes, and we're getting a single expression profile for those monocytes. And then after we've done that, we just run our standard differential expression analysis, the same that we would run if this was bulk RNA-seq. So you can kind of think of Selenics as similar in spirit to cell sorting. So, sorting out our monocytes and then running a bulk RNA-seq experiment on those sorted cells. And the reason that Selenics takes this particular approach is because pseudobulk methods generally outperform other methods on various benchmarks. And they have a number of properties that help to explain this. One particularly important one is that it doesn't treat cells from the same sample as independent. So remember, we're taking those 212 monocytes from the previous slide, all those expression profiles, and collapsing it into just a single expression profile. And as a result, our sample size is the number of individuals that we've actually sampled, which is reflective of where our actual biological replication is in the experiment. So I think that gives you a nice sort of very high-level overview of what Selenics is trying to accomplish and do with your data. So we're going to jump into the hands-on portion of the workshop now, where I'll also fill in some of the details of how exactly do we go from the count data that you get from your friendly neighborhood core to being able to run these cell type specific pseudobulk differential expression analyses, which is really a mouthful, but hopefully you sort of understand what we're getting at for those. Oh, yeah. One other point here is that what we're doing here is quite similar in spirit to something that we actually do for bulk RNA-seq as well, where if you have replicate samples from the same individual in bulk RNA-seq, the best advice for how to treat those replicates is just to average them, within the same sample. So it's fairly similar to what we're doing for this approach. So the first thing that I want to say is that I really want this workshop to be as interactive as possible. So I'm going to be asking you guys to write in the Zoom chat throughout the workshop. And there'll also be opportunity for asking questions. You can post them down in the chat, or during the exercises, you can unmute yourself and if you're comfortable, and ask them there as well. And my first question for everyone is, how experienced are you with analyzing single-cell RNA-seq data? Do you have a little bit of experience, quite a bit of experience, or maybe this is the first time you've done it? So let me know down in the Zoom chat. And the reason why I asked this one is just so I can make sure to pitch the workshop at an appropriate level, and also make sure that I'm giving enough time for the exercises. So I see some of you are already answering that down in the Zoom chat. I really appreciate that. Please let me know if you haven't had a chance to yet. Great. Some experience working with a bioinformatician. A lot of people, first time. Great. Glad you're here. Awesome. So really appreciate that, guys. Super helpful for me. My next question for all of you is, do you have a single cell RNA-seq data set that you're either trying to analyze right now or that you would like to analyze? So this could be your own data set. It could be a collaborator's. This could also be a publicly available data set that you're interested in exploring. If that's you, please let me know down in the Zoom chat. And the reason why I asked this one is just so that I can make sure to follow up with you after the workshop and make sure that, if you need any help getting your data into the platform or with your analysis in general, that you're getting that help. And that's really one of the take-home messages for today is that I'm your go-to person here at HMS for Selenics support. So definitely don't hesitate to reach out to me either, through email or you can book a Zoom with me. And, if you need any help getting your data into the platform or you have another special request, and I'll make sure to circulate this information after the workshop as well. So, so you guys have that. Okay, so when you first logged into the platform, you landed here on the data management module. So it's worthwhile me just giving a very high level overview of what we're looking at. So on the far left here are the navigation controls for the 4 different modules of Selenics. You don't need to click through them now. We're going to be going through them during the workshop. Just to the right of that is the project list. So this is the list of projects that are available to you. If this is your first time in Selenics, you're just going to have the COVID-19 workshop data set in there. If you're an existing user, you might have additional projects here. And if that's the case, go ahead and select the workshop COVID-19 data set. And when you do that, over in the project details tile here, you're going to see that this data set has 3 samples in it. And those 3 samples are from this paper here, which is on SARS-CoV-2 associated MISC, or multisystem inflammatory syndrome in children. And the authors of this paper report on 3 PBMC samples that were isolated from children who were infected with COVID-19 and subsequently developed MISC. 2 of the samples are from patient P13 and P14 when they were in the acute phase of the disease. And the 3rd sample is also from patient P13, but was collected during the convalescent phase, so during recovery. In terms of the tissue type, PBMCs for this data set, I suspect most of you have some familiarity with them. So very high level, these are an immune cell fraction that are isolated from the blood. So they have various immune cell types in it. And they're widely used in multiple fields, given how easily accessible they are and easy to isolate. In terms of what you need to know about the disease, MISC, not very much. So just very high level. It usually develops 4 to 6 weeks after SARS-CoV-2 infection is characterized by a marked inflammatory state in various organs, and it can be quite serious, particularly because of the cardiac symptoms that can develop. And the authors of the paper essentially state that our understanding of this disease is fairly poor, and so they want to characterize how the immune system is behaving in it. So if you haven't managed to log into the platform or access the COVID-19 data set yet, no need to panic. You still have a few minutes while I go over the next module. But if you have, I have an action for all of you, which is to go ahead and click the Go to Data Processing button in the top right here, making sure you have that, the workshop data set selected. And once you do, you're going to land on the data processing module. And I'm just going to give a very high-level overview of what exactly happens here. So this is a 7-step pipeline that gets sequentially applied to your data set. The first 5 steps are filters. So they're removing empty droplets, dead cells, doublets, etc., from your samples. And then after that, your samples are integrated together, adjusting for batch effects between your samples. And then a PCA dimensionality reduction is performed. And that reduction then gets used as input to the last step, configure embedding, where either a UMAP or TSNE is generated, so for those pretty plots, as well as where automated clustering is performed. And there are a number of settings that can be adjusted for all of these steps. And just to note, for the first 5 steps, if you adjust settings, you need to make sure you click the manual radio button before it will let you adjust the thresholds. And my only request is that just for the workshop, please just leave the settings the way they are. Don't rerun data processing. And that's just going to make sure we're all looking at a similar embedding and that you're not waiting for it to rerun. In terms of what the data processing module looks like, as I mentioned, it's a 7-step pipeline. You can navigate through those steps either with the drop-down in the top left or with the navigation arrows in the top right. And we've really tried to make the platform as biologist-friendly as possible. So there's a number of explanation tooltips throughout the platform. And these will give details on exactly what's happening on a given step, as well as what methods are being used. And they'll often provide links to additional reading materials on those methods as well. So definitely do check those out. They can be quite useful for understanding exactly what's happening. Finally, in the top right here is the pipeline status indicator. And it should look like this for all of you, just indicating that the pipeline has been fully run. Okay, so before we jump into our first exercise, I'm just gonna give a live demo of the data processing module and all the steps, just to give you a bit more details on it. So I'm going to be using this data set here. It's different from the workshop data set, so don't be alarmed that it looks different. So I'm going to click Go to Data Processing, and I land on the classifier filter. And the goal of this filter is to remove droplets that don't have a cell in them. So these are empty droplets. And the way this filter works is for each droplet, it runs a hypothesis test, asking whether or not it's empty. And so you end up with a false discovery rate for each droplet. And then any droplet that has an FDR value higher than the threshold here is discarded from your sample. So this particular data set has 2 samples in it. And what we're looking at here in this plot is called a knee plot, and it's showing the distribution of UMIs. So up here is the droplet with the largest amount of UMIs, and down here is the droplet with the fewest. And all the droplets in orange and some of the ones in gray are what's being discarded from the sample. And if you're not familiar with what a UMI is, it stands for Unique Molecular Identifier. And it's essentially the number of unique mRNAs that were sequenced in your droplet. So it's sort of this little trick where prior to any PCR amplification steps, a unique barcode gets added to every mRNA. And then that way, after all the PCR amplification steps, anytime that same barcode is found, it can just be treated, as there's a single read there. So that's how we get these UMIs. So our second filter is the cell size distribution filter. And this filter is also for removing empty droplets. The distinction between this one and the first filter is that here you can set a hard threshold on the minimum number of UMIs that you want in your sample. And because this filter overlaps substantially with the first filter, it's disabled by default, but you can enable it by clicking this button here. And sometimes it will make sense to do this. The first filter, this classifier filter, sometimes it can be overly conservative. And so if later on when you're exploring your data, you find that maybe you have a cluster of cells that has a very low number of UMIs, but otherwise doesn't seem distinct. It shares marker genes with other cells or with other clusters. You might want to come back here and enable this filter and adjust your threshold so that you can filter those out. The third step is the mitochondrial content filter. And this takes a common approach to filter out dead or dying cells. So the idea here is that a dying cell will have little holes in its membrane that the cytoplasmic mRNA leaks out of, and that mRNA that's inside of mitochondria will be relatively better retained in the cell. And so what this filter does is it looks for cells that have an outline high percentage of mitochondrial reads, and then it removes those from the sample. So everything higher than this red threshold line. Step number 4 is the genes versus UMIs filter. And what we're looking at here in this plot is the gene counts on the y-axis. So that's the number of genes that have at least one read in the cell. And that's being plotted as a function of the total number of UMIs in the cell. And you expect to see something similar to what we see here for these 2 samples, where as you sequence a cell to a greater and greater depth, so more UMIs, you detect more and more genes. And then anything outside of these red threshold lines is discarded from the sample. And this is a filter where if you do have a substantial number of cells outside of these red threshold lines, it can be worthwhile to investigate what those are and determine if you do in fact want to be filtering them out. So you could, for example, disable the filter and see what new cells show up. And the most common example that I'll see of this will be a group of cells down here. And you can think of these as low complexity cells. So for a given sequencing depth, a given number of UMIs, there's relatively few genes that are detected. So, these cells are producing a bunch of mRNA, but they're for a relatively small number of genes. So that's why you can think of them as low complexity. And there'll be an example of that in the workshop data set today. Step number 5 is the doublet filter. So for each of the droplets that get sequenced in your data set, there's sort of 3 main cases. The first is that the droplet is empty. And we've already gotten rid of those in the first 2 filters. The second case is that a droplet has exactly 1 cell in it. And this is the ideal case. These are the cells that we want to analyze. And the third case is that a droplet has 2 or more cells in it. And we also want to get rid of those. And that's what this filter does. And the way it does this is it trains a machine learning model that comes up with a probability for each droplet of being a doublet, as well as a classification threshold, this red line here. And then any cells that have a probability higher than that red threshold line will be discarded. And because the threshold value itself is part of the output of the model, this is not typically a filter where I recommend changing that threshold. Okay. So step number 6 is data integration. So we've finished filtering out each of our samples, and now we need to combine those samples. And when we do that, we need to adjust for batch effects between our samples. And the goal here is really to make it so that the same cell type in our different samples will show up together in our data set. And this is taking a little bit to load, but there's a number of methods available to you to perform this data integration. You can select from those here. And there's some settings that you can adjust for that as well as for the subsequent PCA reduction that takes place on your data set. And there's also a couple of plots here that can be useful for evaluating your integration. So for example, we can look at the frequency plot here, which is showing us our automatically detected clusters. And we can see that both of the samples in this data set have cells contributing to all of the clusters. And if we don't see this, we want to investigate further. So, maybe there's a biological reason that it makes sense that one or more of your clusters doesn't have cells or many cells from one of your samples. Alternatively, there could potentially be a quality issue with one of your samples. So you would want to see if you had some other evidence for that as well. It also can sometimes be worthwhile trying one of the other integration methods to see if you get a similar result as well. Okay, finally, step number 7, configure embedding. So this is where our embedding is generated. So either UMAP or TSNE. You can adjust some settings for that, as well as for the automated clustering that takes place on your data set. And the main one that you will sometimes adjust, or that I would sometimes recommend adjusting here, would be the resolution of the clustering. And the idea here is that if you increase the resolution, you're going to get more clusters in your data set, and vice versa if you decrease it. And sometimes it will make sense to do so. You might, for example, find that, in one of your clusters, there's sort of like a region of it that strongly expresses certain genes, but not in another region. So you might be able to come back and increase your resolution to have your clustering reflect that observation. Okay, great. So that's it for the demo. So we'll jump into the first exercise for today, where you're going to look at the three samples in the COVID workshop data set, and you're going to assess the quality of the samples. So specifically, you're going to look at whether they have a similar proportion of dead cells, of doublets, as well as you're going to evaluate the integration. So to do this, you're going to go to data processing, then either using the navigation arrows or the drop-down menu, go through each of the steps in data processing, and look in detail at step number 3, the mitochondrial content filter, step number 5, the doublet filter, as well as data integration, step 6. And once you're done, please give me a done one in the chat so that I know how everyone's progressing. And this shouldn't take you too long. I'll give up to about 5 minutes. And if you have any questions, again, feel free to either post them in the chat or unmute yourself and you can ask them as well. Okay. Okay, so there's a question from Jahan. I know that there are some tests to assess clustering resolution / stability. Is there anything like that in this platform? So great question. Unfortunately, that's not available in the platform. One thing that you can always do is you can always download the Seurat object for your data set. And then you could, if you have an R package that you're interested in looking into that, you could run that outside of the program as well. So to do that, you would just go to data management, go to your data set, click download, and download the processed Seurat object. One thing that I just forgot to mention in the last step, configure embedding, is that there's a number of plots here that can be quite useful for assessing the quality of your data set. Um, so you can plot a bunch of quality metrics on your embedding. So for example, the fraction of mitochondrial reads or doublet scores or the number of genes you're detecting or the number of UMIs. And that can be quite useful because oftentimes when there's quality issues in your data set, they'll show up in a cluster of cells. So it's really good to look out for that. Great. So I see some of you are starting to write done one in the chat. Thanks so much. Uh, Very appreciated for just keeping track of how you guys are progressing. So please continue to do so. Okay, so quite a few done ones in the chat. Really appreciate that again. Thank you so much. So that's 5 minutes. So I'll move on to the answer for this exercise. And it's worthwhile just sort of reflecting a little bit on the goal of this exercise. It's not too uncommon that a user will ask, I have a sample, a data set, and one of my samples, I'm concerned about the quality of it. Do you think I should exclude it from my analysis? Um, and so that's really the goal of this exercise is to give you some of those tools so that you can evaluate that for your own data sets. Um, so looking at the three samples from the COVID-19 data set, and the first thing I had you look at was the proportion of dead cells. So this was step number 3, the mitochondrial content filter. And one of the things that stands out for this is this middle sample, where the histogram looks quite a bit different. If we look a bit closer, we can see that this is mostly being driven by a scaling in the y-axis. So there's a thin green line here, so quite a large percentage of cells that have a very low mitochondrial percentage. But in terms of the actual shapes of the histograms themselves, They all have a median value of about 7%, and maybe 5 or 4% of cells have that median value. Um, and in terms of the percentage of cells that are being filtered out, they're all about 10%. So nothing that concerns me looking at this filter. The thing that you would want to look out for here would be a rightwards shift in one or more of your samples. So if one of your samples had, in general, a higher percentage of mitochondrial reads, that could indicate a potential quality issue with that sample. So the second thing that I had you look at was the proportion of doublets. So again, this middle sample here doesn't look like the other two. And I think the reason for this is just that there's quite a bit fewer cells in this sample as compared to the other two. So if you'll remember, this is training a machine learning model. And I think just because there's less overall data for that process, there's a lower confidence for this middle sample. And so that's what's driving this image. But it's nothing that gives me any particular concern looking at the plots. And to be frank, there's not much that I would think to look out for in these plots. One thing that you can look out for, like I mentioned, is looking at the doublet scores plotted on your embedding. And in this case, we see that there aren't any clusters of cells with high doublet scores. So this is reassuring. Finally, I asked you to evaluate the integration of this data set. So we can see that all three of our samples are represented in all of our automated clusters. We can see this both in the frequency plot as well as on our embedding. And so there's nothing here that would make me want to consider potentially excluding one of our samples or wanting to look more into whether there's a quality issue with it. So let's explore this data set a little bit closer. So when I first looked at this data set, the thing that stood out to me is that two of the samples are from the same patient. One when they're in the acute phase of the disease, and one when they're in the convalescent phase of the disease. And the colors of your samples might look different. They probably will. But in this case, so the three clusters that best fit, or it seemed like a reasonable starting point to me to see if there's any clusters that increase in proportion as this patient 13 recovered. And these were the three clusters that best fit that criteria for me. So, going from blue to yellow, relatively small to much more. And so I wanted to know, what are these three clusters? And that's going to motivate our next exercise. To answer this, we're going to use the data exploration module. And this is going to be where we figure out what our clusters are, where we annotate them. We're going to be doing that by finding marker genes for our clusters, which are just genes that distinguish one cluster from the rest of our cells in our data set. This is also going to be where we perform those cell type specific differential expression analyses that I talked about at the beginning of the workshop. This is really where you're going to be probably spending the majority of your time in the platform, looking for interesting biological observations and finding interesting things about your data set. This slide here is just a reference for this next exercise. of some common cell types that you would expect to see in a PBMC data set, like the workshop data set. I don't expect you guys to be intimately familiar with this, so I've included some, common marker genes for these cells. And I'll put this up partway through the next exercise if you want to give yourself a bit of a hint. So here I've just gone ahead and provisionally noted the three clusters that went up in proportion as patient 13 recovered. And I'm going to show you guys how to annotate one of those clusters, this cluster 12 here. And then for the exercise, you're going to annotate the two remaining clusters. So I'm going to jump back into Solenix. And this time I'm on the workshop data set. I'm going to go to data processing. I'm going to jump down to data exploration, where in the top left here is my embedding. Here is that cluster 12 that I'm going to show you how to annotate. Just to the right of the embedding is the cell sets and metadata tile. This is where you're going to find your automated clusters. You can select them, see how many cells are in them. This is also where you're going to find custom cell sets that you can define. I'll show you how to do that. Anytime you see one of these little eyes in the platform, if you click it, it's going to use that information to color your embedding. So here you can see that I've used my custom cell sets to draw a beautiful chicken in this drumstick shaped cluster here, and I'll show you guys how you can do that. This is also going to be where you find any sample level metadata. So just for example, the sample identity of your samples in your data set, or in this case, the MISC status of our samples. So whether they're in the convalescent phase or the acute phase. And then finally, this is also where you're going to be able to access automated cell type annotation using the up here in the Annotate Clusters tab. This uses sctype. And what you do is you select the cell, the cell type or the tissue type of your data set. So in this case, it would be immune system. And then it's currently implemented for either human or mouse data sets. And my general view on automated cell type annotation is that it can be a really useful starting point for your annotation. But you do want to know how to manually annotate your data set still, just so that you can, evaluate those annotations. They'll often be incorrect. So moving over to the right here is the genes tile. And the default view here is just the list of genes in your data set. So again, if we click one of the eyes beside one of the genes, it's going to plot the expression of that gene up in our embedding. This is also where, the differential expression tab here, this is where you're going to, when you select this first radio button here, that's going to be what you use for finding marker genes for your cluster. So when you want to annotate them, figure out what type of cell they are. Whereas the second radio button here, that's what you're going to select when you want to perform those cell type specific differential expression analyses. So, treatment versus control in monocytes, what's different about them? Finally, in the bottom tile here is where we have our heat map. And the default view for this is just we have our automated clusters, and we have the top few marker genes for each of those clusters. And this can be customized quite a bit by adjusting the settings here. And we'll do that for our last exercise today. So I mentioned that I'm going to show you guys how to annotate this cluster 12. So I'll show you two different approaches you could take. One of them is just to come down into your heat map, head on over to your cluster 12, and then head down to this block of genes where it shows the top few marker genes for that cluster. Here we have HBB, HBA2. If you don't know what these are, you can come up here. We can search for them. We can, click the eye to plot their expression. We see, it's strongly expressed in that cluster 12. We can also click the name of the gene. It's a link, and that's going to take us to the gene cards entry for that gene, where we can see that this particular gene is a hemoglobin subunit. So suggesting that these are red blood cells. If there aren't enough marker genes in the heat map to figure out what type of cell you're looking at, that's when you would want to come up to the differential expression up here. And again, you're going to select this first radio button in order to get, because we want to get the full list of marker genes for this cluster. So we're interested in our cluster 12, and we're going to compare to all other cells in our data set. There will be times when you want to compare to just a single other cluster. This is really useful when you're trying to distinguish between two closely related clusters. So for example, these two up here. But in this case, we're just going to select all other cells, and we're going to keep all of our samples for this comparison. We run that, and we get our full list of marker genes. Again, we can plot their expression. This one here, ALAS2, highly expressed in our cluster 12, minimally everywhere else. We can go see what it is. Quite a bit more obscure, 5' aminolevulinate synthase 2. If we read a bit more, we can see that this particular gene catalyzes the first step in heme biosynthesis. So, quite certain that these are red blood cells. It's not always going to be this easy to annotate your clusters. It's been made deliberately easy for this example. It's going to take a lot of searching around often. Some marker genes aren't going to make sense. You're not going to be able to figure out, why they're a marker gene or what type of cell they should belong to. But don't despair. Okay, great. So now that we know what this cluster is, we want to record that information somewhere. So what you're going to typically do to do that is just come up to your automated clusters, go down to the appropriate cluster 12 in this case, and click this little edit button beside it, and then just rename it. Another thing you might do, I'm just going to really show you just so that you're familiar with it, is you can come and click this little lasso button up here, and then you can click and drag to select a group of cells. And again, we can give it a name, and that's going to pop it down into our custom cell set. So you can see we've now added those RBCs to my beautiful chicken drawing. Okay, great. So now that we have a question. Where can we find how many cells are in each cluster? Great question. So again, if you just click the radio button or the check box beside one of your clusters, it's going to show you how many cells are selected. And that can be, you can do that for your samples as well, or any sort of metadata. So great question, thank you. So now that you know how to approach annotating your data set, I'm going to have you annotate these two remaining clusters that went up in patient 13 as they recovered. So to do this, you're for the olive cluster 7 up here, I'm going to steer you to use the approach where I showed you, where you go and you look at the top few marker genes in your heat map, maybe search for them, go see what they are, plot them, try to figure it out that way if you can. Whereas for the blue cluster 4, just so that you get a bit of experience using the different parts of the platform, I'm going to recommend that you come up to the differential expression tab, select that first radio button, etc., to get the full set of marker genes, and then explore those. If you, once you're finished those first two clusters, let me know with a big done to in the chat. If you do have some extra time, one thing you can try to do is try to figure out what distinguishes this olive cluster 7 that you've just annotated from the nearby yellow cluster 2. And I'll give you guys about 10 minutes for this. If you have any questions, again, please post them in the chat or feel free to unmute yourself as well. And there's a question from Rachel. If you have two clusters selected, is it the total of both? And yes, yeah, that's the case. Great question. Thank you. So there's about 5 minutes left. I'm just going to put up that reference slide in case you want to give yourself a little bit of a hint. So we have a question in the chat. Rachel's wondering if you hover over the heat map on the left, does it link to the list on the right? Great question. That sounds like it would be an awesome feature. Unfortunately, it doesn't. So yeah, if what you would do is, you find the gene that you're interested in, try to remember the acronym and then come up here and search for it in the gene list.
Okay, thanks, Alex.
Oh yeah, happy to help. If you guys have any more questions, definitely feel free to post them and um, let me know when you're finished. Give me that big done too. But we'll go for another few more minutes. I see some of you are finishing up. Uh, really appreciate you letting me know. Definitely keep posting those done tos in the chat once you're finished. Okay, so quite a few done twos there. Great to see you guys are uh, really doing well. That's oftentimes the exercise where people get pressed up for time. So no, don't be concerned if you weren't able to finish. Um, so we'll move on to the answer now for this exercise. So hopefully you were able to come up with something similar to what I have up here in bold, which is that the olive cluster 4 is a cluster of B cells, and the blue cluster 7 is a cluster of monocytes. Um, so now that we have figured out what these clusters are that went up in proportion as patient 13 recovered, let's do a little bit of interpretation. The first cluster, the one that I annotated, cluster 12, was red blood cells. And this isn't particularly interesting. This is a cell type that's actively excluded from the PBMC fraction. So, maybe this represents just a bit of overzealous pipetting. Um, our next cluster, cluster 7, was a cluster of B cells. And this particular cluster of B cells expresses high levels of immunoglobulin G antibody genes. And you might have had a chance to look at this if you had a bit of extra time. That's in comparison to the nearby yellow cluster 2, which has much higher levels of immunoglobulin M antibody genes. And the fact that this IgG producing B cell cluster increased in proportion with recovery, this is consistent with what we know about the typical course of a COVID-19 infection, where immunoglobulin G antibody levels rise later on in the infection. Finally, we have our cluster 4, which was CD14 monocytes. And this same observation, that this cluster increased in proportion with recovery, this is what most interested the authors that originally reported on this data set. And so they went on to further characterize these cells using a much larger set of samples. So what we're going to do for our next exercise is just make it really clear how this cluster is changing in proportion between our two time points. This particular plot here, this frequency plot, is not the best way to evaluate that, especially when there's large differences in the cell number between our samples, as there is in this case. We really kind of want to flip this plot on its head, where instead we're looking at our three samples, and we're looking at the proportion of our different cell types in each of those samples, and that way it all adds up to 100%. So to do that, we're going to use the plots and tables module. So fairly self-explanatory. There's a number of standard plots that are available to you. Importantly, you can fully customize these plots so that you can hopefully get the styling just the way you need to, either for a publication or for a presentation. And there's a number of formats that you can export those plots in as well. So as I mentioned, we're really going to zoom in on those CD14 monocytes. So to do this, we're going to start by creating two custom cell sets. So the first one is we're just going to duplicate our CD14 monocytes. So you're going to come and you're going to select cluster 4 here, and then you're going to click this second button up here in 1A. And that's going to create a new cell set by combination. So it's combining any clusters that we've selected. In this case, we've just selected a single cluster, so it's just going to duplicate it. So give that an appropriate name, like CD14 monocytes. And then still with that cluster 4 selected, come and click this last button in this row up here, which is 1B. And that's going to create a new cell set by complement. So any cells that are not currently selected. Um, so give that one an appropriate name, like all other cells. And then once you've done that, jump down into the plots and tables module, where you're going to open a frequency plot. And then under the select data heading, you're first going to group by sample, and then you're going to subgroup by your custom cell sets in this case. So they'll not Louvain here. Take a look at that plot, see what you make of it. And then when you're finished, please give me a big done 3 in the chat. This one should be pretty quick. I'll give up to about 5 minutes, and let me know if you have any questions. So we'll go for up to about 2 more minutes or so here. I see a lot of you are already finished. Again, really appreciate um, all the done 3s. Okay, great. So I think uh, that's pretty much everybody. So you guys are rock stars as usual here. So we'll move on to the answer for this one. So hopefully you were able to come up with something that looks similar to what I have up here, where we can see that um, in our two acute samples, there's a fairly substantial lower proportion of our CD14 monocytes, particularly in the case of sample from patient 13. And this is in comparison to that same patient during the convalescent phase. So this would be reasonable. Oh, yeah, Rachel, you have a question?
Yeah, I was going to type it in, but I thought it might just be easier if I ask it. So the
Sure.
way that mine came up, it's like the opposite um uh in order, the opposite order, like the
Ah,
convalescent is first and then the acutes are second. But it looks like overall, I think it's the same pattern, although it looks a little different. So I was just curious if there's something that I did like,
yeah, no, great question. So, what you can do to rearrange the order of your samples, this is slightly obscure, so it's really good of you to ask, is in data exploration, under the samples heading here, you can just drag and drop your samples, and that will rearrange the order of them. And that should also apply for the plots and tables as well.
yep, that looks like it worked. Thank you.
Great, awesome. Thanks for the great question. Okay, so this would be reasonable evidence here to want to further pursue what else is different in this cell type, just like the authors that first reported on this data set did. Um, and so that's exactly what we're going to do for our last exercise of today, where we're going to perform one of those pseudobulk, cell type specific differential expression analyses, that big mouthful in these monocytes. And so the way we're going to do that is we're going to do that in data exploration. Um, and the first thing that you're going to do is you're going to come up to the differential expression tab. And this time you're going to select that second radio button, right? So that's for comparing, between treatment and control samples or whatever your particular case is within a specific cell type. So in this case, that specific cell type is going to be cluster 4, our monocytes, and we're going to compare our convalescent sample to the rest of our samples. Um, you're going to run that analysis, and that's going to give you your list of differentially expressed genes. And the next thing that we want to do is we want to filter that list so that we're only looking at significant genes. So to do that, you're going to come and click this advanced filtering button, number 2 here. And then under the presets dropdown, you're going to select significant. Go ahead and apply that. And that's going to make it so that you're only looking at genes with an FDR of less than 0.05 in your table here. And then once you have that, the next thing that we want to do is we want to take those genes and overwrite our heat map with them. So to do that, you're going to click this little checkbox up here, number 3. That's going to select everything. And then on number 4, heat map, you're going to click heat map and then overwrite. Might take a minute or so to grab all the data, but that's going to put those genes down in your heat map. If you're with me so far, you're doing good. Just a couple more steps. The next thing that you want to do is add the samples metadata track to your heat map. And that just lets us see, which sample is expressing a given gene. And to do that, you're going to click this little settings cog, and you're going to toggle on the samples metadata track. And then finally, the last thing you're going to do is number 6 here. You're just going to come and click the hide button beside the all other cell sets that you created in the last exercise. And that's going to make it so that you're only looking at those CD14 monocytes, both up here in your embedding, as well as down here in your heat map. Once you have your platform set up in that way, go ahead and read the couple of sentences up here at the top of this slide. Look at these genes and see what you make of them. And then give me a big done for in the chat. And like always, if you have any questions, don't hesitate to ask. And um, I'll give up to a maximum of about 10 minutes or so. So we'll go for about another 6 minutes or so maximum. Again, really appreciate all the dones in the chat. Really helpful to see how everyone's progressing. Okay, so I think that's pretty much everyone there. So again, great job. In terms of um, the couple of sentences I had you look at up here, the first gene was FCGR1A, which is forms part of the receptor for the immunoglobulin G. And we can see in our heat map that it's consistently expressed in our two um, samples from the acute phase, and then it's gone right back down in the sample from the convalescent phase. And this same observation was reported by the authors who collected this data. So we've gone ahead and reproduced that finding. The second gene that I had you take a look at, RNase 1, is not something that was originally reported on. I put it in there myself. I thought it was fairly interesting. It shows quite a very similar pattern of expression. It's up in the acute phase and comes back down with recovery. And I thought it was interesting because this is an RNA degrading enzyme, so potentially representing part of the antiviral response. And it would make sense that it would, come back right back down with recovery. So that really wraps up the hands-on portion of the workshop. And the only thing that is still outstanding for me to cover is to give you a bit of information so that you can know how to upload your own data set to Solenix. So in terms of where you're going to do that, that's going to be in the data management module. And to do so, you're just going to click Create New Project, and then Upload Project, give your project a name, and then you're going to add data to your project, number 3 here. And in terms of the most common formats that Solenix accepts, I'll go over that in the next couple slides. But before I do, I just want to comment on adding metadata. So when you click this button, what it's going to do is it's going to create a new column in your table. So for the workshop data set, it has a column called MISC status, and then values of either acute or convalescent. We could have clicked the button again and added a column called patient ID and had P14 or P13 as values. And this is really important to add before you process your data set, because you're going to be able to access these annotations when you're exploring your data, when you're performing your cell type specific differential expression analyses. These annotations are also going to be available to you in various plots as well. So definitely do make sure you add that to your data set. So in terms of the most common types of data that Selenics supports, so things you can add when you click this Add Data button, the most common file formats will be files coming from 10x Chromium or saved Seurat objects. There's a number of other formats that Selenics accepts, but I'll just cover these because they're sort of the most common. So for 10x Chromium, you're usually going to be uploading either the filtered or unfiltered count matrices that are coming out of Cell Ranger. And if you're getting this from a core or someone else, they'll usually be stored in a folder named raw feature BC matrix. And for each sample in your data set, you need 3 files, a features.tsv file, a barcodes.tsv file, and a matrix.mtx file. And these can be gzipped or not, doesn't matter. You take those 3 files and you put them in a folder with the sample name as the folder name. So for example, we have our sample1 folder, and we have those 3 files in it. And once you have this for all your samples, you're just gonna, select them all and drag them into the pop-up that comes up when you click Add Data. And all this information is in that pop-up as well, so you don't need to remember it. The second most common file format that you might use is going to be a saved Seurat object. So this is going to be a .rds file. And the idea behind this file format is that it lets you run all of the steps of data processing outside of Selenics. And then you can just use Selenics to take advantage of the point and click data exploration and plotting functionality, as well as all the downstream analyses. So this is a really good file format if you're working with a bioinformatics collaborator. They can generate this file for you. You can upload it to the platform, and you can really continue your analysis of your data set right at the point where they left off. You don't have to redo a bunch of things. In terms of the requirements for this type of, for a saved Seurat object, it needs to have a column called samples in the metadata that indicates, what sample all the cells are from. It also needs to have the raw counts in the RNA assay. And then it also needs to have a PCA as well as either a UMAP or TSNE reduction. And if you have those 3 things, then Selenics is going to automatically detect any clusterings that have been saved in your object, as well as any annotations of those clusterings that have already been performed. And it's also going to automatically detect any sample level metadata that's stored in that object as well. And for both these file formats or any other file format, if you need any help getting your data into the platform, you're not sure if your data is supported, definitely reach out to me. I can often make things compatible, even though maybe they didn't initially upload correctly. So just summing up for today, I hope that as a result of the workshop, you have a better understanding of how Solenix can support your single cell analysis. That you've gained a bit of comfort for exploring a data set in Solenix. And most importantly, that you know where to turn to if you need any help, getting your data into the platform or with your analysis, which is, reach out to me. So with that, I just want to thank everybody for coming, and all the fabulous questions, and the excellent level of participation. So thank you. And if anybody has any remaining questions, now's definitely the time. I'm happy to stay for a while.